The theme of our research is the search of somatic embryogenesis regulators in plants. Somatic embryogenesis is a way of plant regeneration comprising the development of embryo-like structures from vegetative tissues. It is widely applied in biotechnology for transformation and propagation of plants, and also it is very useful in fundamental study because it can serve as a model to study regeneration and embryogenesis. In this picture, you can see the somatic embryos of Pisces abies, and as you can see, they can develop into new plantlets, and they then can grow and develop into adult plants. So how can you stimulate somatic embryo development? The traditional way to do it is to use phytohormones, mainly auxins and the cytokinins. So using this way, you take the plant explants and, for example, leaf explants, and put them onto the medium, in vitro medium, with different combinations of phytohormones. Usually, first of all, auxin is applied, and callus can be developed from the explant. After that, you usually should remove auxin and or reduce its concentration, and after that, embryos, somatic embryos, or can appear, or shoot regeneration can start. Recently, a new way of stimulation of somatic embryogenesis appeared. It includes the usage of so-called morphogenic regulators. They can use instead of phytohormones or together with them. Usually, morphogenic regulators are transcription factors that participate in somatic embryogenesis, and they can induce this process via direct inductors of embryo or shoot development. For example, here you can see GRF GIF1 dimer, its effect on the shoot regeneration, and you can see that when you transform the explants with a construction containing GRF GIF dimer, such calli can regenerate more effectively. In this picture, you can see the application of agrobacterium containing plasmid with other genes for other morphogenic regulators genes, such as Wuschel, shoot meristemless, baby boom, and so on. And so when you apply agrobacterium on the part of tissues, of plant tissues, the transformed cells can regenerate new plant. So we study somatic embryogenesis and we search for morphogenic regulators using Medicago truncatula as a model object. Medicago truncatula is a model legume, and it has different lines, which has very different capacity for somatic embryogenesis. It can be useful for search somatic embryogenesis regulators. We started our search from VOX family. VOX proteins are homodomain containing transcription factors, which can regulate many aspects of plant development, like for example, root or shoot development or embryo development too. We found that overexpression of one of VOX genes, VOX91, can stimulate somatic embryogenesis in Medicago truncatula. So here you can see wild type calli and the calli with VOX91 overexpression, which have much more green somatic embryos on the surface. This experiment was performed on the embryogenic line. So here you can see that the calli of this line have embryos, although not so much as in this condition, with this genotype. But Medicaca and Catla also have a non-embryogenic line, for example, 1081. Now we're checking the capability of VOX91 to stimulate somatic embryogenesis in lines and species which are not able to form somatic embryos. We transformed a non-embryogenic Medicaca line 1081 with constructions for VOX91 expression, and such Kali in about 30%. About 30% of Kali gave somatic embryos, whereas control Kali almost never could give an embryo, any embryos. Now we try to apply these results to other species, for example, Pisum sativum. So now we are in progress, and we transformed the same constructions. We transformed sparkle cultivar of P, and we already see such some differences between control and sample too. So this is a work of Sviatoslav Baltin, my student, and also we made CRISPR mutants of VOX91, and it appears that plants with loss of function of VOX91 are almost completely sterile and have malformed inflorescences and pods too. This data suggests that VOX91 definitely participates not only in somatic but also in usual zygotic embryogenesis too. Another member of VOX family, VOX2 gene, as you can see here, is able to stimulate callus growth. So this is control Kali, and these are Kali with VOX2 overexpression. They are much bigger, and you can see it here. In some cases, the weight of such Kali is more than 1,000 milligrams, which is unusual for such Kali. So this is the work of Elizaveta Krasnoperova. She has a poster in this conference. You can check it out too. We performed transcriptomic analysis of Kali with VOX91 or VOX2 overexpression, and we found that rather a lot of many differentially expressed genes in both cases. About 3,000 of genes in VOX, differentially expressed genes in VOX91 overexpressing Kali, and about 1,700 in VOX2 overexpressing Kali. So what is interesting is that the differentially expressed genes in both kinds of Kali are highly overlapping. With significant overlap, which is highly unlikely to be a coincidence. The most of these genes, common differentially regulated genes, are activated by VOX91 and repressed by VOX2. So we performed gene ontology enrichment analysis of these genes activated by VOX91 expression and suppressed by VOX2 overexpression, and found a lot of pathways related to somatic embryogenesis. As you can see here, so this is somatic embryogenesis pathway, regulation of abscisic acid, gibberellic, and gibberellin biosynthetic process, and also regulation of seed germination, seedling development, photosynthesis, and so on. So we can suggest that VOX2 and VOX91 have partly opposite effect on gene expression during in vitro culture, and VOX2 stimulates callus growth, whereas VOX91 stimulates opposite process, quite opposite process, somatic embryos development. We have checked the influence of overexpression of several genes on somatic embryogenesis. And also the VOX family in medicated cattle is rather big. So our next step will be to check what influence do other VOX genes have on somatic embryogenesis capacity. This is the work of Daria Yakovleva. VOX genes are known to be regulated by peptide phytohormones, so-called clepeptides. For example, such regulatory loop are known for short apical meristem, where VOX gene Wushel is regulated by clavata3, and also in root apical meristem, where VOX5 is regulated by CLE40. We also checked if some CLE peptides exist that can regulate expression of VOX genes, and by means of that, regulate somatic embryogenesis. To date, we found that a CLE16 gene its overexpression reduces the amount of embryos per callus. This is our first target for editing. So we plan to obtain the plants with loss of function of these genes and to check their embryogenesis capacity, so they can be more capable to regeneration, we suppose. We search for somatic embryogenesis regulators among NFYB genes, so-called LEC1 type NFYB genes. Why we are so interested in them? It's because LEC1 gene, or leafy cotyledon, is famous and widely known as a master regulator of embryogenesis, and its overexpression in Arabidopsis can stimulate somatic embryo development of ectopic somatic embryos. Its orthologs in Medicago truncatula, NFYB10 and NFYB3 genes, are highly expressed during somatic embryogenesis, specifically during somatic embryogenesis. So you can see here, these are from 4th to 6th week of cultivation. Somatic embryos start to appear in the calli of another embryogenic line, 2HA, and only at these stages, the expression, and only in embryogenic line, the expression of B10 and B3 is elevated. Unfortunately, overexpression of these genes, B10 and B3, did not stimulate somatic embryogenesis in our hands. So all types of calli had the same somatic embryo, mean somatic embryo number per callus in comparison with control. But we also checked the interactions between NFY B10 and 3 and other members of NFI groups. These are NFIA and NFIC proteins, which these three groups of proteins are known to form heterotrimer consisting from NFIB, NFIA, and NFIC. We found supposed partners of YB3 and YB10 genes among NFIA and NFIC families. And also we found that this NFIA can also have elevated expression during embryogenesis. So we plan to check if the overexpression of these genes, NFIA genes, can stimulate somatic embryogenesis, maybe together with NFIB and NFIC genes. This is our working scheme. I think that it will be actual for several years, several future years. We search somatic embryogenesis regulators among cleptoptides, Box genes, and NFI genes. We check their influence on somatic embryogenesis intensity, and we hope that our data will help to develop new and fast plant genome editing and transformation protocols. So at this slide, you can see our group and also our collaborators and the scientific foundations which give grants for this research. And thank you for your attention.